Hi everyone. Let us now start with the next topic of general microbiology, and that is the bread and butter of a microbiologist, a pathologist, or anyone sitting in the lab, and that is the world of microscopes. This is exactly why this subject exists, right? So, before I start with the different types, there are certain common features that every microscope has, and the main purpose of an existence of a microscope is that its ability of magnification. You can compare it actually to a camera. Like in camera, you have lens systems with which you can zoom in 2x, 10x, and so on. Similarly, in microscopes, you have lenses with which you can magnify, and that is how we can see the different bacteria, viruses, kidney biopsies, and so on. But what's also important that despite having a lens system, a beautiful picture always comes when you add some filters to it. So similarly, in a microscope, for you to see a better picture, you need to create a contrast. It's something similar to how you edit your pictures and change the brightness and the contrast, etc. Same way, if I want to see a more detailed image, I need to add a contrast, and that is with the help of adding some colors or dyes. That is why in pathology it's said to be the world of pink and blue. In microbiology, you have Gram stain, Zn stain. Till the time you don't add some dyes or colors, you won't be able to enhance the features. Lastly, the third point about a microscope is the resolution. Taking you back to your physics days, resolution is the ability to differentiate two points as separate. Now over here, if I have made these 2 points, you can see them as two separate points because your eyes have that resolution. If I would have made them very, very close, as close as this, probably you wouldn't have been able to visualize them as two separate points. So that means that what is the resolution of the eye? It is 0.2 mm. If I want to see two dots as separate dots, the distance between them should at least be 0.2 mm, then my eyes will be able to see them as two separate. If the distance is less than 0.2 mm, then my eye will perceive it to be a single dot. So the resolution of the eye is 0.2 mm, and now keep it 0.2 but keep changing the unit. The resolution for a light microscope is 0.2 microns. Resolution for an electron microscope is 0.2 to 0.5 nanometers, which tells me precisely why viruses are only seen on an electron microscope, because the size of a virus is in nanometers. You will never see a virus on a light microscope, and of course never with your naked eye. So viruses are only seen on an electron microscope. That is why knowing the resolution is so important. So all in all, the features of a microscope include the lens system making the magnification, the resolution ability, and finally the contrast by dyes. Now let's start with the real microscope which you find in every lab, and that is a light microscope. I've told you it was given by Sir Anton von Leeuwenhoek, but he gave you the simple microscope. The microscope that you're using nowadays in your lab is known as a compound microscope, and the reason is that it is going to have two set of lens systems. As usual, you will visualize with me that there are a couple of lenses and hence it's compound. So the purpose of calling it a light microscope has to be that it has a light. So you can very well visualize that there is a light source over here, and this is something very similar to a tube light or a bulb that you have. Actually, it's a kind of a bulb, so nothing very fancy in terms of the light source. I can say that this is a transmitted light, like the light in this room is coming towards me, so that's a transmission. In the air, it is getting transmitted. So the light from this bulb is actually going to get transmitted. Transmitted where? It is going to go, and over here, this is known as the stage. What is a stage? A stage is where the hero is going to perform. So who's the hero of this story? The hero is going to be a slide. That is where your specimen is, be it a kidney biopsy, brain biopsy, a TB bacteria, or maybe a virus. That slide is always kept on the stage because the hero has to be kept on the stage. Now the problem is that the light that I had kept, the light can go in air in any direction, right? But I want my light to come as a spotlight on my hero. That is, I want the light to come on the slide. That is why I have kept an additional structure over here, and that is known as a condenser. Firstly, they ask you the location of the condenser, and this is going to be present below the stage. And next they will ask you that what does condenser do? Of course, the name tells me that it is going to condense the light, but they ask you many variations. Does it condense the light in the air? Does it condense the light onto the lens? Does it condense the light onto the slide? Of course, onto the slide, so that is where your hero is, and you want to create a spotlight. So imagine light entering your eyes or light entering the camera. There is some adjustment you can do, like you have a diaphragm in the camera which you can close and open so that the amount of light is varying. Similarly, this condenser is also going to have inside it something that is known as a diaphragm. Finally, if I trace the light, I can see that the light was going everywhere, but the condenser said no, you only have to go to this hero, that is the slide. And after this, the light will now start going to the lenses. I have one set of lenses over here. This is known as the objective lens. The reason for calling it objective lens is because it is near to the object of the slide. And there are many objective lenses, because of course you want to zoom in maybe 4 times, 10 times, 40 times. So you have many lenses, and you can keep revolving them depending on whichever lens you want to see. So for example, if you're rotating your head, your rotation is happening on your neck. Same way, if you're rotating these objective lenses, the rotation is going to happen on the neck piece. So this area where you're going to rotate everything is going to be known as the neck piece. And this is where the light is passing. And finally, after passing through this, it will come to my eyes. I am sitting over here as a pathologist or a microbiologist, and because my eyes are peeping into the lens, this near the eyes is referred to as the eyepiece. So now I have two lens systems. The near one to the object is objective lens. The near to the eyes is known as the eyepiece lens. Once you have done all of this, you will say that, ma'am, if I am sitting over here and looking at the slide, I will have a different vision. Maybe you have spectacles or lenses and I have a normal vision. So we are not going to keep the slide at the same distance. I might want to keep the slide closer to my eyes. You might want to keep it away from your eyes, because we all have different vision. That is why moving this stage up and down, adjusting the stage, is very important for every person to have its own vision adjustment. For that, you have a big knob over here, and this is known as the coarse adjustment, because you want to do coarse, many centimeters up and down. If you want the stage to move, that is coarse adjustment. Versus if you say, no ma'am, almost our vision is the same, but still you also have 6x6 vision, I also have 6x6 vision, but still there is some blurred image that is coming. You are looking into the microscope, you feel that no, the slide is looking slightly blurred. If 1 mm also the slide would have moved, that image would become clear. So you want to do a very fine adjustment, few mm. That is done by the smaller knob over here, which is called fine adjustment. So these are all the parts of a light microscope. But before we move forward, let's go through a quick 20-second review of what are the parts and how the labeling can be asked. So of course, what's the most important in a light microscope is going to be the source of light, and it is going to be transmitted light. Of course, light can travel in all directions, but you want all of the light to come on the stage. The reason being that the hero of your story, that is the slide, is kept on the stage, and you want the spotlight to be created on the slide. Well, that spotlight will be created by the next structure, which is known as a condenser. The condenser is going to be present below the stage, and it is going to help in keeping your light onto the slide. After that, your light is going to pass through all these lens systems. So there are two lens systems. The ones that is next to your eyes with which you are peeping in, that lens is known as the eyepiece lens. And the one that is closer to the object is known as the objective lens. Because there are many lenses, they will be rotating around this area. And this rotating area is known as the neck piece. Now, if you want to do variations in this stage and you want to move it up and down, you have one very big adjustment known as the coarse adjustment, or this big knob over here. But if you just want to move it by few mm, then you have a smaller knob over here, which is known as the fine adjustment. Now, another thing that I want to highlight over here is the concept of numerical aperture, but this of course is only important for INI CET aspirants. For that, I first want to orient you that I have made a system of lenses on the top and I have made the slide on the bottom. So of course, light is going to pass through this, and that is how you will see the slide. So maybe you have some kind of a specimen over here, and this is where light is passing through. Also, what you can visualize is that as I was going from one side to the other, the magnification is increasing. So over here, if I look at this lens, this has a magnification of say 4x. The next lens is going to have a magnification of 10x, and the next lens is going to have a magnification of 40x. These are some random numbers of magnifications that I've given to you. So what did you notice? Actually, when the light is passing through, it is trying to create an angle. If you look carefully, there's a cone that is created. So you will say that, yeah, there is an angle that is created between this lens and the light. An angle is definitely created. I'm going to divide that angle into half. The reason for dividing that angle into half is that half the angle is known as theta. So what is that angle? It is the angle that is created, not the angle, but half of the angle that is created between the light as well as the lens. This half angle is known as theta. And if you look carefully, as the magnification of the lens is increasing, 4x, 10x, 40x, don't you think that that angle is also increasing? The value of theta is increasing more in the next one. The value of theta is increasing more in the next one. So all all in all, I can say that now I can explain to you what is numerical aperture, because that is a formula. It is n sine theta, where n refers to refractive index and theta refers to half the angle. So I understood that as the lens magnification is going to increase, I understood that the value of theta is increasing, and if the value of theta is increasing, automatically numerical aperture increases. So taking the starting and the last point tells me that as the lens size or the magnification increases, there is going to be an increase in the numerical aperture. Of course, this is a little bit of physics and maths involved, but I think this is more than enough to know for INICET. Now coming to the basic concept that light is actually passing through two lenses, the objective as well as the eyepiece. This means the final light and the final magnification that I see is going to be a combination of these two lens systems. So what are these lenses and what are their magnifications? First, let's learn those. For the eyepiece lens, because I have only one lens and there is no variation, the magnification is always 10x or 10 times. But for the objective lenses, I have a system of 4. So scanner is known as 4 times magnification. Low power is 10 times magnification, so high power will be higher, that is 40 times magnification. And if you add a drop of oil and try to look at whatever specimen you have, now the magnification with that lens will be 100x. So remembering it more like a sequence is 4, 10, combine the two or multiply, that's 40, and 100. So I can say 4, 10, and then add zeros in front of them, so that becomes 40 and 100. Repeat with me: 4, 10, 40, 100. 4 is going to be known as the scanner, 10 is low power, 40 is high power, and 100 is oil immersion. But I'm not interested. When I'm looking at the microscope, I'm just looking at a total. So if someone asks you that what exactly is the total magnification, you will multiply both of them. I will multiply this objective lens and the eyepiece lens. So that is what you do. You multiply objective multiplied by eyepiece, and that will give you the total magnification. Let's try to do this with the help of some examples. I've got you a fill in the blank over here. These are all the objective lenses that I have written for you, and they have different magnifications. But for the eyepiece lens, we know that the magnification will always be constant in every situation, that is 10x. So what is the total going to be? If you're using the scanner, then the total, see, multiply for eyepiece, it's always 10x. So this means you just look at the objective and you have to multiply it by 10. So for scanner, it is going to be 4 multiplied by 10, the total magnification is 40 times. For low power, the total magnification is 10 into 10, that is 100 times. For high power, the maximum magnification is 40 into 10, that is going to be 400 times. And for oil immersion, it is 100 into 10, and that is going to be 1000 times. That tells me the most important question and PYQ in the exams, that the maximum magnification of a light microscope is in front of you, and that is 1000x. But so many students mug it up. What if I change the question? If I've asked you the maximum magnification of a light microscope, it's 1000x. But if I ask you the maximum magnification of the objective lenses, and now it is 100x. So please read the question carefully. Well, of course, on that note, you have finished your entire light microscope, and now you're ready to take on to its counterpart in a slight variation that is known as the dark field microscope. No points for guessing that the photo is going to look very dark over here. The reason being that I have used a special light called the reflected light. So before I move forward, I just want to tell you the basic difference, that in a light microscope, I have used a transmitted light like a bulb, whereas in dark field microscope, I'm going to use a reflected light. But what's becoming a little, it should generate curiosity, and that is that I am using light and still, ma'am, there's darkness despite using light. Yes, and that is how I'll tell you with the help of a video. But before that, this kind of a microscope is used whenever you want to visualize something very thin, which you can't visualize otherwise on a light microscope. For example, flagella. Flagella helps in what? Motility. So can I say that for motility, I can, for testing motility of very, very thin structures, like let's take the example of a very thin bacteria, that is spirochete. No points for guessing that spirochete is spiral. Example of spirochete is the one that causes syphilis, sexually transmitted disease. So it's very, very thin. If you want to see the movement of this spirochete, you will not be able to see it under a normal light microscope. You will have to use a dark field microscope. So whenever you have to see something very, very thin, and for thin bacteria, if you want to see their motility, you are using a dark field microscope, and this is how it looks like. The name does its own justification. Dark field tells me that the background over here has turned out to be dark, and of course, because of that, the organism is going to get highlighted. How is it that I'm using a light and I'm still getting darkness? Now let's understand this. So on one side, I'm going to explain to you the light microscope, also known as bright field microscope, and on the other side we have the dark field. But when I see these manufacturings, both of them look exactly the same. In both of them, I'm going to have a light source. The light is going to go first via a condenser. The condenser will send the light onto the stage where my slide or sample is present. And then the light is going to go to the lens system, of course, first to the objective lens. So overall, everything looked the same. Giving you a quick recap, what happens in a light microscope is that the light is going to go and is going to hit the object, that is the specimen. After that, the same light will now travel through this entire objective and eyepiece lens system, and the final photo that you get is a lot of light. It's very bright. You can see the organism getting highlighted. That is why it's a light microscope. On the other hand, I realized that I have the same parts in a dark field microscope with one difference. The same condenser that I had put up over here, I have created a stop in the center. A stop means a kind of an area which will not let the light pass through. So this area is known as a stop, or some people call it a central aperture. This is very important to know because now see how the light passes through it. See, the same light is passing through, but I noticed that there is a certain area in the center where the light has not passed through. So the final outcome that I get is going to be a cone of light, but is it a complete cone of light or is it a hollow cone of light? So if you look carefully, there is no light in the center. So this of course is a hollow cone of light, and this is exactly the reason why there is a darkness that is created, and you can see how the background has turned out to be dark. Repeating once again, whenever you want to see, and this is going to be your take home message, whenever you want to see the motility of very thin bacteria, you are going to use the dark field microscope, because they will ask you if they don't add all of this. If they say, I just want to see the motility of bacteria, then even your light microscope is good enough. Motility, actually, we visualize it on a day-to-day basis. If I want to see, is a bacteria moving or not? Like let's say Vibrio cholera shows you shooting star motility. It moves like a shooting star within seconds, very fast. I will look at it under a light microscope. But if I want to see the motility of a very thin bacteria, and an example of that happens to be spirochete, then that's so thin that I can't look at it under a light microscope, and then I will take the help of a dark field microscope. This is going to be that buzzword in the paper which will help you solve the MCQs. Coming to the third microscope, and that is the phase contrast microscope. The name itself is telling me that there is going to be a contrast in the phase. So if I look at this, I have the same light, I have the same condenser system, then I have my sample, then I have the objective lens, and over here with the eyepiece, you have the eyepiece lens. Everything is similar to a light microscope, but I've added two things. One thing that is added is known as an annulus ring. It's like a ring, so some people call it annulus condenser because it's in the condenser. Some people call it annular diaphragm because condenser and diaphragm mean the same. And some people also call it an annular ring because it's in the shape of a ring. And you can see it's just next to the condenser. So I call it an annulus condenser to remember it. Another thing that is added right near to your eyes is referred to as the phase plate. And now let's know a brief about what is happening over here. So over here, the purpose of having different phases is that this is done whenever you're dealing with different refractive indices. There will be always some structures, like if I want to see the internal part of a structure, internally there might be a different refractive index of the cell, externally there might be a different refractive index. So whenever I'm dealing with different refractive indices, I'm trying to create different phases. Just giving you a slight overview of what happens from physics point of view is that initially when you have started, you switched on the light. Initially, can you see that when the light waves have started going on, all the light waves are in the same phase, because if you see the waves, they are all coming parallel, they are all in the same phase. So I'll repeat it once again. You can see in the form of these curvatures, their phases are the same. But as soon as they hit the specimen, because the specimen has different refractive indices, I see that these phases have changed. Now see, this phase has shifted here and this has shifted there. Now it's no longer in the same straight line like it was. Can I say that there has been a considerable phase shift in the light? And now I know the reason. The reason for this phase shift is because this specimen over here, this cell had different parts, different organelles, different structures. So they had different refractive indices. They have split the light. They have changed the phase of that light. And now when this kind of a light passes through the phase plate, your phase phase plate is going to change the intensity, and then what it will show you is going to be different intensities of the light, and that is known as phase contrast microscope. So what is it used for? Number one, it is used for seeing the live organisms, because now I notice that because you have changed the phase of light, you don't need to add any dyes and you don't need to add any colors to create that contrast. That contrast has already been created by whom? By the change in the phase of the light. Now you understand phase contrast, that you are creating a contrast by doing a shift in the phases of the light, and hence a phase contrast, which means I will be able to see unstained specimens or live organisms without adding any dye. Also, if you want to visualize internal structures of a cell, you want to know the inside organelles, this is a really good microscope, because with different refractive indices, all the internal details will be shown. So that tells me that whenever you are dealing with live organisms, you can see the motility over here also. So now if I have to do a recap, I realize that if I want to see the motility of organisms in general, I don't know what is the structure and the shape. Most common way of looking at the motility is the routine light microscope. But when I am dealing with certain thin bacteria, there are two options that I have. Either I can look at the motility under the dark field microscope or under the phase contrast microscope. So both are useful, but if I have to select one in the exam for thin organisms, the preferred one is dark field, and that is performed more commonly. So please read carefully. In general, motility is seen on light microscope. For thin bacteria, it is seen on dark field and phase contrast, more commonly on dark field. Now let's move on to the next microscope that we have, and that is going to be the fluorescent microscope. Now let's move on to the next microscope, and that is the fluorescence microscope. I think it is pretty obvious that you're going to get some shiny fluorescent pictures over here. When I look at it, it looks like any other regular light microscope. So internally there have to be some changes for me to make it a fluorescent microscope, and let's see what the changes are. So the first change is that what I'm going to see as an end product will be a nice shiny fluorescent picture, but internally what's happening in the microscope is what I have to know. Firstly, the kind of light that I use is not a regular light, it is going to be a mercury lamp. The reason for using it, that a mercury lamp is going to create ultraviolet light. I hope you all remember your basic physics chemistry that ultraviolet light is not visible. Till now, everywhere I was using visible light, be it a light microscope, dark field microscope, phase contrast microscope, but here I am using ultraviolet light, which is not in the visible range. I will have to make it in the visible range later on for me to see it. So over here I can see that the ultraviolet light is passing through, and the first thing that it comes across is an excitation filter. Now excitation means it's going to excite the light, so it will have a different aperture, it will have a different wavelength. How do I decide which filter to use? That will depend on what kind of dye you have added on the specimen. Yes, here is where the slide is. Maybe this is a case of a pathology slide or a kidney biopsy, because in that you were using fluorescent microscope. Maybe this is a skin biopsy. Over there you do fluorescence and you see the fishnet and other patterns on bullous disorders. Maybe this is a microbiology slide where you're trying to see TB. Whatever may be the specimen, you will have to add some dyes, and the name of these dyes have been known as fluorophores. You can easily understand that fluorophore means some fluorescent dyes will be added. So if I'm adding a green color dye, I will have a different excitation filter. If I'm going to add a blue color dye, I will have a different excitation filter that the company and the protocol will be given to me. Now once the light has passed through the excitation filter, can you directly bring this light towards your eyes? Of course not, because UV light is going to be in the non-visible range. I will first have to make it visible. So what I do, I make this light first strike on a dichroic mirror. A mirror will always reflect the light, and that is what has happened. The light has now been sent towards the sample, and and in this sample what I have added is a dye or a fluorophore. Now the purpose of adding this dye is that this dye is going to change the wavelength. The wavelength that was coming initially is a shorter wavelength, and after it strikes the sample, it is going to be converted to a longer wavelength. So now the light that is going to go back, if you look carefully, the light that is going back is going to be of a longer wavelength, but that has to pass through the emission filter. The name only tells emission. This will not let any other light pass. This will only let the emitted light pass. So the light that has been emitted from the specimen, only that can pass through it, and now it is going to come to my eyes, and my eyes will then see that beautiful fluorescent picture. So I know that the four main questions that you get is what kind of light do we use? Is an ultraviolet light? What is that fancy mirror used in a fluorescent fluorescent microscope. It is a dichroic mirror. The principle is that a shorter wavelength is converted into a longer wavelength, and what is added to the specimen are many colors of fluorophores that are dyes. So your next question is, what dyes are added? Depends what sample I'm dealing with. If you're dealing with a case of TB, suspected case, then the fluorescent dyes that you use are going to be auramine and rhodamine. Only for the students who are targeting the conceptual and the Central Institute exams, you should know that auramine and rhodamine are going to bind to the mycolic acid of TB, which is a part of their cell wall. But please note, I'm saying auramine and rhodamine. I'm not saying rhodanine, because in pathology you had studied that rhodanine is going to be a stain for copper, whereas rhodamine and auramine is going to be for Mycobacterium TB. The next one is going to be malaria. For malaria, the fluorescent dye is acridine orange. And which part of malaria does it go and bind to? It binds to the DNA of the malaria. Lastly, for fungus, we have calco-flower white, which is so obvious. The floor tells me it's a fluorescent dye, and calco means which part of fungus is calco-flower white going to bind to? It is going to bind to the chitin of the fungus, which is present in the outer part of the fungus. So overall, I have a couple of pictures over here. I would zoom in and want to show you that in the first photo, these are all the rod-shaped TB organisms that I'm seeing, means I've used auramine, rhodamine. In the next picture, you can see the different ring form of malaria. You can see the schizont of malaria. You can see that banana shaped gametocyte also over here. This means this is a case of plasmodium or some case of malaria over here, and the dye that you must have used for malaria is acridine orange. Coming to the next one, and that is going to be a case of calco-flower white. Why did I say that? Because over here I can see that the dye has gone and gotten bound only to the outer part of the fungus. Why are you calling it a fungus, ma'am? Because I can see these tiny buds coming out of it. Budding is a feature which is always a kind of a division that the fungus undergoes. So now I know that this calco-flower white is going to be the one, it's a fluorescent stain binding to the outer wall or the chitin of the fungus. These are definitely asked, but when you're talking about fluorescent dyes, how can you forget the fluorescent dye that you used in pathology? When you were doing a technique called flow cytometry in hematology for detecting all the CD markers, you all used a dye, and that was known as FITC. This was also fluorescent, fluoroisothiocyanate. So see, the fluoro word does not leave you in path and micro both. Finally, you have done all of fluorescent microscope that you had to know. But first, I would want to show you a small image, because that could be a way of asking the question in the exam. So a quick video to understand a fluorescent microscope. You know that the light source is a mercury lamp or an ultraviolet light which is not in the visible range. So the first thing that you have to do is to make that light pass through something known as a dichroic mirror, which is going to reflect that light downwards onto the specimen. After the specimen changes it into a longer wavelength, you can see how the light has gone all the way back, and after going back, the final image that you see onto your eyes is going to be a fluorescent image. So the basic functioning of a fluorescent microscope requires two things. One is going to be an ultraviolet light via a mercury lamp, and the second is going to be the working of a dichroic mirror, which is very essential for the working of this microscope. Finally, ending it with questions important for INI CET exam. You can very well know that there are some structures which are autofluorescent, which means that till now I was saying whenever you add a color or a dye, it is only then that the fluorescent color will come out, like a green color. But there are some structures which will shine even without adding a dye, and that is how you should be in life. No one should be adding that extra element to you, and you should still shine on your own. So structures which are shining on their own, number one is going to be, I call them the Spora brothers. The reason is because they are named similarly, cyclospora, isospora. You put them under ultraviolet light, they will start shining. You don't need to add any auramine, rhodamine, acridine orange, calco-flower white, nothing. They will shine as soon as you put ultraviolet light on them. You will see them shining on their own. So autofluorescence. The next one is going to be formalin. Yes, formalin is also autofluorescent. That is why a very, very important practical point over here, that in skin biopsies, which you do for which kind of disorders? Usually it is done for bullous disorders. Giving you an example of pemphigus. In pemphigus, you see the fishnet pattern I taught you in immunofluorescence. Whenever you know that a dermatologist is doing a skin biopsy and he wants that you should look at that skin under the fluorescent microscope, tell the dermatologist, do not send me that skin biopsy in formalin, because formalin, as soon as I will look at it under the ultraviolet light, formalin will only start shining. I will never ever be able to know whether the skin is actually showing me the fluorescent reaction or not, because formalin will make everything shine. Please, in this case, if you want me to look at the skin biopsy under a fluorescent microscope, send it to me in normal saline. Normal saline does not show any fluorescence, but fluorescence is shown by formalin. So if you put anything in formalin and then you look at it under a fluorescent microscope, the formalin will automatically make it shine, and then everything will start looking fluorescent in that situation. The third autofluorescent thing is known as NADPH. Now, if you remember, NADPH is a molecule that is formed in the G6PD pathway, which means that if you have G6PD enzyme working in your body, that is when NADPH will form, and people who don't have G6PD or are G6PD deficient are not going to have any NADPH formed in their body. Why am I teaching you hemat all of a sudden and G6PD anemia? Is because that NADPH is autofluorescent, which means people, if I take your blood, and you and I'm assuming and I hope that all of us have G6PD enzyme working, this means all of us have NADPH in our blood. If I take a drop of your blood and I look at it under ultraviolet light, my blood will shine. Why? Because I have NADPH. But people with G6PD deficiency anemia and who don't have any NADPH in their body, if I take their blood and I look at it under ultraviolet light, they will not show me any shine or any fluorescence, which means NADPH is fluorescent. And that is why for G6PD deficiency anemia, we do a test with blood that is known as fluorescent spot test, which tells me that if you see the fluorescence, this means your NADPH and G6PD are working fine and you are good. But if you don't see any fluorescence, this means your NADPH and G6PD are not working fine and you have G6PD deficiency anemia. So done with fluorescent microscope, we move on to the next one, a rather rarely asked one called interference contrast microscope. Again, you're trying to create a contrast, and that will only be when you want to peep inside the cell. You want to see the organelles inside the cell, because you know that, okay, everything will have a different phase, everything will have a different contrast. You are trying to interfere inside. That is how I learned it. Because you want to interfere inside the cell and the organelles, you will use interference contrast microscope. That's not it. It also helps you know certain chemical constituents. You want to know about the lipids, the proteins, the nucleic acid. That interference also the interference contrast microscope tells you. Well, the last microscope that you have to do now, the one that looks like a spaceship ready, rocket ready to launch because it's closed from all sides, and that is electron microscope. I taught you electron microscope was E for E, that is, it was discovered by Ernst Ruska. So now what is an electron microscope? I will teach you simultaneously as I teach you a light microscope. So let's start right from the day when a pathologist is getting a slide or a microbiologist is getting a sample from the clinician. If the clinician wants to get a light microscope done, this means the clinician will send the sample in which fixative? 10% neutral buffered formalin. But if the physician wants that you should see it under an electron microscope, maybe the physician is expecting some virus. Viruses are seen only on electron microscope. Maybe the physician is dealing with a renal biopsy and over there you have to see everything under electron microscope. Then the sample will be sent under the fixative 2 to 2.5% glutaraldehyde. So the fixative of light microscope is 10% NBF, and for electron microscope it is 2 to 2.5% glutaraldehyde. So the sample has come to you in this fixative. Now what are you going to do? You are going to take that sample out and you are going to embed it. In a light microscope, you put it inside paraffin wax. I'll show you a picture. That is what you did in case of a light microscope. This, for example, was a brain biopsy. You took the brain biopsy and you have put it inside what? You have put it inside paraffin wax so that you can preserve it for many years, like 5 years, 10 years. And when you have to transfer it on a slide, you will cut it and then you will transfer it where? You will transfer it on a glass slide. I think everyone knows this about a light microscope, but the story is different for an electron microscope. The embedding is done in resin. You do not use paraffin wax over here. Paraffin wax doesn't go well with electrons later on. So you embed it in resin and then you cut it onto a slide which is made up of copper or metal. So this is what resin is. This is how you've embedded it inside resin. This is your sample, and when you have to cut it, you are going to cut it out and you're going to put it on a slide which is made of metal, and usually that slide is made of copper. All these things you're doing because in electron microscope you are dealing with electrons. So maybe the electrons will not work well with wax. Electrons will not work well with glass. So for electrons I need to embed it in resin and I need to put it and cut it on a metal slide, that is copper. Finally, coming to what are you using? Of course, in a light microscope I'm using transmitted light like a tube light or a small bulb, and that light can easily travel in air, so the medium is air. But electrons can't travel in air, because in electron microscope, of course, the source is electrons, and that is going to travel only in vacuum. Now you understand why this was enclosed from everywhere. I need to create that vacuum, otherwise the electrons will be lost. So for the electrons, a vacuum has to be created. Finally, I come down to that what is the resolution. This you've already done earlier, that for a light microscope the resolution is 0.2 microns, whereas electron microscope is obviously having higher resolving ability, and it can see something as tiny as 0.2 to 0.5 nanometers. How we had learnt it, light microscope 0.2 microns, electron microscope even higher, that is 0.2 nanometers. Finally, coming to the lens system, which I think you've studied with me. Under the light microscope, you had one lens near the object, so objective, one lens near the eyepiece or eyes, and that is eyepiece. Same way, in the electron microscope, one lens is near the object, and that is objective, but there is no exact lens near your eyes, because if you look at this, where are you peeping in from? It is closed from everywhere. So ultimately, whatever lens system is there is going to project it onto a computer screen, right? So I can say that yes, there is going to be an eyepiece lens, but it is going to project it. That is why rather than calling it eyepiece, I call it a projector lens. Please note, near the in light microscope it is object and eyepiece. Near the object, near the eyepiece, I have two lens systems. In electron also there are two lens systems. Near the object it is objective, but then I'm not peeping in because it's closed, so it's going to be projected, and that is why it is a projector lens. Also note, because you are dealing with a beam of electrons, this entire closed space is kept under magnetic influence. That is why these lenses are also known as the magnetic lenses. Well, giving you some higher information over here is that electron microscope is further of two types known as SEM and TEM. SEM is known as scanning electron microscope. So scanning means that what are you trying to do? I'm only trying to scan, ma'am. I'm only not trying to see the details of anything. I'm trying to see the superficial, like something like a rapid revision. You're just trying to see the superficial concepts and quickly revise your syllabus, right? So I can say that I'm only trying to see the upper details of anything. I'm not trying to go deep inside the organism or the bacteria. I'm only trying to visualize the surface details, which means that I will be able to cover, like if you're doing rapid revision, in 1 hour you will be able to cover 2-3 chapters. Same way, if you're only looking at the surface details, you will be able to visualize a lot of sample that will be viewed in a very less time. Like in rapid revision, you can do a lot of chapters in very little time, right? That means what kind of electrons am I using over here? Over here I'm using something known as the scattered electrons. That is why you called it scanning electron microscope, because you have used scattered electrons. They are going to get scattered and they will only tell you the surface details. The kind of image that they finally form on the screen is a three-dimensional picture, because on the surface they are able to tell you the entire 3D model of that organism. On the other hand, you are dealing with something known as TEM. TEM word tells me that you are going to transmit the electrons through and through. The kind of electrons that you are using are transmitted electrons, because let's say if this is your sample, if the electrons just touch the surface and get scattered, that's a scanning electron microscope, because here you are trying to scatter the electrons, so only the surface is being visualized. But if this is my electrons and the electrons pass through and they get transmitted, that is a transmission electron microscope, because now by transmitting the electrons, I can show you the inner details, which means that now I'll be able to visualize all the internal details. It's something like doing your main videos. In your main videos, you're trying to look at every concept, every detail, so it is going to take time. In a particular 1 hour, you will not be able to cover 10 chapters. You'll probably be able to cover 2 pages. So this means very little sample is viewed in a less time. And because you're going right inside and you're looking at every layer, you are trying to only make a 2-dimensional picture over here. Let's understand this also a little better with the help of a video. So let's have a look at what's happening in a scanning electron microscope. As I told you, there are scattered electrons which will only show you the surface details. So as you can see over here, it is only the surface that is being projected onto the screen. Are you able to look at anything inside or any internal details? Not at all. So as they said, in a scanning electron microscope, the kind of electrons are going to be scattered electrons. What you shall get to see are only the surface details, and you will thereby, you can see how quickly the entire surface was scanned. So you will be be able to see a lot of sample very quickly. Let's have another view of exactly what has happened over here. So this is a scanning electron microscope. You can see how the electron beam is going to come, and it is only the surface that is quickly scanned. Now you know why I call it scanning electron microscope. Now compare this with the other type, and that is TEM. TEM refers to transmission electron microscope, where the electrons are going to go through and through. And that means that here I have an electron beam, and how that electron beam is going through and through, and I can see every little internal detail that I have in that organism or cell. That is what is transmission electron microscope. One quick view for you once again. You can see the electrons going all the way through and through, and that means all the internal details will be visualized over here. Of course, with that, you wrap up all your microscope questions, but now we go in for some MCQs to test whether you're able to apply that knowledge. So scanning electron microscope is used for what? When you're scanning, so you're only trying to look at the surface structure, and that is the answer over here. Coming to the next one, maximum magnification of a light microscope is 1000x. No two thoughts on that. In a light microscope, what is the function of condenser? Firstly, condenser was something present below the stage. So below the stage means it's going to put that spotlight on the stage. That's your sample. It is going to focus the light where? It is going to focus the light on the sample. The next question is a microscope that exposes the specimens to ultraviolet light. I know that's a fluorescent microscope anyway, so I mark that as an answer, but still they say it forms an image emitted at a different wavelength. Yes, the principle is that shorter wavelength is converted to longer wavelength, everything going for fluorescent microscope. The next image is also of a fluorescent microscope, and they say which of the following structures is required for this image. So we know that no, this is not a dark field microscope, this is not to do with phase plate, it's not phase contrast, there is nothing cathode ray tube needed. I need a dichroic mirror, because for fluorescent microscope either you need an ultraviolet light and you need a dichroic mirror. Coming on to the next question, which is these days the examiner's favorite. He will tell you to write the sequence of a light microscope without giving you a photo. So a microbiologist intern, I don't know who does their internship in microbiology, but nonetheless, he wants to study cells and microorganism. Again, who wants to do that? But his senior advised him to use a light microscope. What is the arrangement? You know the arrangement. You just need to know how the examiner has asked you. From the eyes to the light source. He's not asked you from down above, he's asked you from up to down. So from the eyes you have to start. Right next to the eyes, what will you have? Of course, you will have the eyepiece lens over here. Then you come from the eyepiece lens, you will have another set of lenses over here, which is known as objective lens. Then you will have the stage, and below the stage you will have the condenser. In case they would have asked you from down up, then your answer would have become condenser, then objective lens, and then eyepiece. So it's so important to know from where to where has been asked. Over here, from eye to light source has been asked. That is why option C is the answer. Well, that's a wrap up to all the microscopes. And now, having done scientist stains and microscopes, you're ready to take on to the real world and start with bacterial anatomy and physiology.